Good morning, my dear students. Today we'll discuss about genetic engineering and restriction enzymes. The biotechnology in the early 1970s was concerned with the production of commercial products generated through the metabolic action of microorganisms. It may be defined as the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. Biotechnology since 1961 has clearly and irrevocably been associated with the study of industrial production of goods and services by using biological organisms, systems, and processes. The biotechnological processes that use microorganisms for production of commercial products typically employs three steps based upstream processing, which involves preparation of a raw material so as to use as food source for target microorganism. Next, fermentation and biotransformation, which also involve growth of the target organism in large bioreactors of more than 100 liters size, resulting in the production of desired compounds like antibiotic, an amino acid, or protein. And lastly, the downstream processing, which involves purification of the product from the cell mass or cell medium. The focus of the research during 1960s and 70s was to improve upstream processing, bioreactor design, and downstream processing to increase yields of the various desired products. The improvement in biotransformation was lacking as this step was the most difficult one to manipulate. Natural occurring microbial strains cannot perform large-scale production of useful commodity. Mutations were used to improve microbial strain, but the level of improvement that could be achieved in this way was usually limited biologically. However, the nature of biotechnology was changed forever with the advent of genetic engineering or recombinant DNA technology. With these techniques, the maximization of the biotransformation phase of the biotechnology process was achieved more directly. Genetic engineering provided a means to create highly productive strain useful for biological production of large amount of useful compounds. The recombinant technology begins with the cloning of specific genes of interest, and gene cloning procedure involves two essential steps, that is, incorporation of the gene of interest into small self-replicating chromosomes called vectors, forming recombinant DNA molecule. For example, insertion of a human gene into an E. coli plasmid and introduction of the resultant recombinant DNA molecule in an appropriate host, for example, a bacterium, for amplification of the gene of interest. This ability of the geneticist to construct desired recombinant molecule at will is the basis of the recombinant DNA technology that has revolutionized molecular biology in the last 3 decades. The first recombinant DNA molecules were produced in Paul Bach's laboratory at Stanford University in 1972. They constructed the recombinant DNA molecule by inserting phase lambda genes into small circular molecule of simian virus 40. Paul Bach was awarded Nobel Prize in Chemistry for this discovery. The expression of human insulin gene for insulin production in bacteria, which seems like a science fiction some few decades ago, was made possible due to development of genetic engineering. The human insulin gene was isolated and inserted into small self-replicating plasmid, forming a recombinant molecule, which was then introduced into bacteria which act as hosts. The host bacterium multiplies so as the recombinant plasmid vector, resulting into rapid amplification of human insulin gene. Today, many human proteins are routinely produced in bacteria or eukaryotic cells growing in culture. Genetic engineering or gene cloning employs certain biological products and biological agents which may be referred to as genetic cloning or genetic engineering tools for achieving its objectives. Some of the most important tools basic to almost all gene cloning experiments are restriction endonuclease, we also call it as molecular scissors, which is used to cut the DNA at specific sites. Then we have vector, which are self-replicating molecule into which the gene of interest or DNA to be cloned will be integrated. And then we have DNA ligase to shield the nicks existing in resultant recombinant DNA molecules after insertion of desired gene. We have host, which is the appropriate organism into which the vectors will be transferred for amplification of gene of interest. Now let us come to restriction enzymes. Organisms contain number of endonucleases which produce internal cuts or cleavage either at only one or both the strands of the DNA duplex. Most of the endonucleases cleave DNA molecules at random sites except for some endonucleases which cut DNA at specific base sequences called recognition sequences or sites. Such endonucleases which recognize specific restriction sites for cleaving DNA molecule are called restriction enzymes or restriction endonucleases. The ability to clone and sequence essentially any gene or other DNA sequence of interest from any species is possible with the discovery of the restriction enzymes in 1970 by Hamilton Smith and Daniel Nathans. The term restriction endonuclease is derived from the Greek word endon, meaning within, depicting restriction enzyme making internal cuts in the DNA molecules. The biological function of restriction endonuclease enzyme is to protect the genetic material of bacteria from invasion by foreign DNA molecules. The restriction endonuclease will bind to the recognition site and cuts its DNA strands, leading to the degradation of DNA. The organism should protect the cleavage sites of their restriction enzymes so that its DNA is not cleaved and degraded, or it would be like committing suicide by the organism. The endonuclease cleavage sites are protected through methylation of one or two nucleotides in the recognition sequence. The methylation of the cleavage sites is catalyzed by specific methylase enzyme, and the methyl groups block the binding of restriction enzyme to the cleavage sites. Now, let us come to the naming of restriction endonucleases. The restriction endonucleases are named by following certain pattern. First, the first letter of the genus from which the given enzyme discovered is written in capital letter, followed by the first two letters of the species name of the organism. The three letters are written in italics. For example, ECO derived from Escherichia coli. Here, the capital E comes from the first letter of genus Escherichia, and CO comes from the first two letters of the species name coli. Similarly, HIN from Haemophilus influenza and HAPA from Haemophilus parainfluenza. Secondly, if the enzyme is produced by a specific strain, a letter designating the strain is appended to the name. For example, ECO-R, as the enzyme is produced by Escherichia coli, strain RY113. In the third point, the first restriction enzyme produced by the Escherichia coli, strain RY13, is designated as ECO-R1. R indicates the first restriction enzyme derived from the particular E. coli strain. If the bacteria strain produces more than one restriction enzymes, the number can be given as 2, 3, and so on. For example, HIN2 and HIN3 for the two restriction enzymes deriving from Haemophilus influenza strain RD. In the fourth point, all the names of restriction enzymes are prefixed with general symbol R. For example, R-ECO-R1, R-HIN2, R-HIN3, etc. This is done to distinguish restriction enzymes from the corresponding methylases, which are isolated from the same bacterial strain. However, the prefix R is not used where the context makes it clear that the enzyme under reference is restriction endonuclease only. Let us come to the types of restriction endonucleases. The restriction endonucleases can be broadly classified into three distinct types: type 1, type 2, and type 3, depending on their composition and enzyme cofactor requirements, the nature of target sequence, and the position of their DNA cleavage site relative to the target sequence. First, let us take off the type 1 restriction enzymes. They are complex, multi-subunit endonucleases that cut DNA at random, far from their recognition sequences, about 1000 base pair away from the 5' end of the sequence TCA present within the recognition site. They have little practical value since they do not produce discrete restriction fragments or distinct gel binding patterns. We have type 2 restriction enzymes. They cleave DNA molecules either within or close to their recognition sequences. These are the only restriction endonucleases which are employed in restriction mapping and gene cloning because they cleave DNA at specific sites. There are more than 350 different type 2 endonucleases with over 100 different recognition sites. Then we have type 3 restriction enzymes. They are large combination of restriction and modification enzymes and cut DNA outside of their recognition sequences and require two such sequences in opposite orientations within the same DNA molecule to accomplish cleavage. They are not used in gene cloning as they do not cut DNA at specific sites and also rarely give complete digest. Then we'll come to production of staggered and blunt ends. The type 2 restriction enzymes commonly recognize DNA sequences which are palindromes, that is, the base sequence in the second half of a DNA strand is the mirror image of the sequence in its first half. The complementary DNA strand also exhibited the same pattern. The restriction sites of most type 2 restriction endonuclease consist of 4, 5 or 6 base pairs which are predominantly rich in GC content. Some restriction enzymes digest DNA at different locations. We call it as staggered cut, which is within the recognition sequence, producing 5' phosphate or 3' hydroxyl extension called protruding or sticky ends. The sticky ends produced due to staggered cuts by specific restriction endonucleases have complementary base sequence because of palindromic nature of the recognition sites. This property can be utilized for the generation of recombinant DNA molecules. The DNA from two different sources, when mixed and digested by same restriction enzyme, produce complementary cohesive ends. Base pairing of complementary ends of different DNA molecules results in the formation of recombinant DNA. Two different DNA, A and B, when clipped by restriction endonuclease BAMH1, produce 4 fragments. 2 DNA fragments derived from different A and B sources of the restriction digest can anneal to one another due to complementary sticky ends to form a new recombinant DNA molecule. Some restriction endonucleases produce blunt ends as they cleave both the strands of the DNA molecule at the same sites. We also call it as even cut. Example is the even cutting of DNA molecules at the restriction sites by Hint2 producing blunt ends. Popular restriction enzymes like EcoR1, PST1 and HAPA2 produce overhang or cohesive ends, while SMA1 and SAE3 generates blunt ends. In order to form a complete recombinant DNA molecule, the existing nicks created have to be joined. This is done by reforming the internucleotide linkage between 3' hydroxyl group and 5' phosphate group in the backbone at the two broken bond sites, which we call it nick. The DNA ligase enzyme from bacteriophage T4 performs this function by catalyzing the formation of phosphodiester bonds. In case of sticky ends, nick is filled by phosphodiester bond formation at the ends of DNA strands that are already held together by the base pairing of the two extensions. The DNA ligase also joins the blunt ends by forming phosphodiester bond between 5' phosphate and 3' hydroxyl groups. This is possible when both ends come in contact when they both bind to the enzyme. Finally, we have conclusion. The development of genetic engineering or recombinant DNA technology has enabled the researchers to isolate specific genes of desired characters from one organism and multiply them in an unrelated host organism. The impact of genetic engineering on the biotechnology has been quite extraordinary. The most effective way of increasing the productivity of organisms before the introduction of recombinant DNA technology in the field of biotechnology was the use of mutation, which was endowed with many limitations. The emergence of genetic engineering technologies offered an alternative by providing rapid, efficient, and powerful means for creating microorganisms with desirable superior genetic attributes. The primary objectives of recombinant DNA technology in identifying and isolating desired genes would not have been successful without the discovery of type 2 restriction enzymes. These enzymes cleave DNA molecules reproducibly into fragments of discrete sizes by binding to specific recognition sequences within DNA molecules. The different tools of recombinant technology enable not only microorganisms but also plants and animals to be genetically modified and engineered to suit our requirements and objectives.